So I'm from Berlin, and I'm in charge of an outpatient clinic, actually for adult immunodeficiencies, where we see many patients with chronic fatigue syndrome as well, approximately 300 per year. And the reason why we see these patients there are that a number of these patients suffer from frequent infections, and we see immunodeficiencies, especially antibody deficiencies, approximately in 25% of these patients. Today I'm going to talk about the studies we perform in CFS on Epstein-Barr virus, and why became we interested in Epstein-Barr virus. Well, a subset of our patients, and that is not only true for Germany, that is also true for other countries, has disease onset with EBV infection. And that is typically a late EBV infection, because when you get EBV as a baby or small child, you usually have few symptoms, but the adults often get quite severe EBV infection. And many patients then have a prolonged infection with severe fatigue, and several patients report that they have a disease which feels like the EBV infection is ongoing, so with recurrent swollen lymph nodes, pharyngitis, fever. We see in a subset of patients elevated antibodies against EBV of IgM type, or against the so-called early antigen. I will go to this point a little bit later. And also in some patients we can detect EBV in peripheral blood. It usually sits in B cells, and we detect elevated levels of EBV DNA. It's a DNA virus. And there's data, especially from Stanford, that some patients improve on antiviral treatment, which is effective against herpes viruses. So what type of virus is EBV? It belongs to this family of human herpes viruses, and that is one that has been very long with us, more than 1 million years. So it's evolutionary, a very old and well-adapted virus. That means it's well adapted to our immune system as well, and can circumvent killing by the immune system. So if you get an infection with a herpes virus, you don't get rid of it. So you have to live with this infection for the rest of your life. However, mostly it's a latent infection, because the virus is so well adapted. It's important for the virus that it makes us not sick, so it usually hides in a non-active form, and can in some individuals, however, reactivate from time to time. And what you all know is the number one member of this family is the simplex virus. Approximately 1/3 of us suffer from sometimes recurrences with lip blisters. This is an infection which most of you have chronically. Another virus most of you also bear is the varicella zoster viruses, the cause of chickenpox and shingles, which may recur in some older people. And then EBV, we don't know that much about EBV, because when EBV reactivates, it doesn't show on the skin like many other of these herpes viruses. The typical symptoms of EBV reaction is sore throat, because it reactivates frequently in the epithelium of the pharynx, and it makes fatigue, a little bit fever. So that is something which may not be obvious for us and for the physicians, and is often misdiagnosed as a common cold. And therefore the data is not clear if EBV frequently reactivates at all, and if it's a subtype of people who suffer from reactivation of EBV. So the primary infection, as I said already, is mostly in early childhood via saliva. So you get it from your parents usually. It sits not only in the B cells in the blood, but as I said, also in the epithelial cell of the pharynx, where it reactivates from time to time. That's normal. That's something you can measure in the saliva. If I would look into your saliva, approximately 20% have evidence of active EBV infection at the moment, which is often asymptomatic, and 80% of the kids are already infected. You can measure this by positive EBV antibody response. And it used to be much higher. So in earlier times, one can assume that all kids had been infected at early ages. But nowadays, where we live under more hygienic conditions and where we no longer chew the food and feed the kid directly, and there are many people who get first EBV infection in adolescence, well known as glandular fever or kissing disease. This is a much more severe type of infection then, and here you see this severe type of pharyngitis we often see, and these adults often have a severe course. They are quite sick, have high fever, feel very fatigued for many weeks, and this can last for months and even years. But usually the adolescent recovers. There are many interesting epidemiological studies about late EBV infection. For example, it's known that it's associated with autoimmune diseases like MS, multiple sclerosis, and SLE. So you cannot develop MS without having been infected with EBV. And if you now go into the adults, approximately 98% are seropositive. In most, it's not disease-causing, but it's a big problem in immunodeficiency. So either if you have genetic immunodeficiency or if you get immunosuppressed, for example, in a situation where you need renal transplantation, then EBV can reactivate because EBV needs to be controlled by the immune system, and then you can get chronic active EBV infection, and that may induce B-cell lymphoma. So here I show you how we usually diagnose EBV. That's important because many patients with CFS get this testing. That is, has been well known for a long time that EBV may play a role in CFS as well. So many patients come to us and say, I have a positive EBV antibody titer, so is this probably cause of my disease? And in many patients, I can say, no, it's completely normal what your antibody response is. So upon the primary infection, we make antibodies, immunoglobulins, specific against EBV. And in EBV, there are two important structures where we test the antibodies against. The one is the surface protein viral capsid antigen called. And in the beginning, you make these so-called IgM antibodies. This is the first immediate type of antibody we make. And then within the first 2 weeks, you start to make IgG antibodies as well. These are the long-lasting antibodies. And then when the virus infects the B cells and the epithelium and starts to make new viruses. So viruses always want to infect cells because they are very simple structured beings. They only have some nucleic acids and some protein, but they need our cell machinery for replication. So when the cells infect, when the virus infects the cells and produces new DNA, then the immune system recognizes the nuclear antigens and makes antibody, IgG antibody against EBNA. So this is what we all have in our serum, the antibodies, which are marker for latent infection. That tells us that we had been infected with EBV probably a long time ago, and that we mounted a normal immune response. What usually is absent in latent infection is IgM antibody, and in addition, we can test in some laboratories the response against the so-called early antigen. The IgG early antigen is also a marker for early infection or reactivation. So coming back to our project, what have we been doing so far? The one is that we wanted to characterize in more detail the EBV-specific B and T cell response in these patients, because that tells us more about the type of infection and if there's probably some deficiency in controlling EBV, because it's already well known that most of CFS patients have no overt EBV infection, as we know from patients who got a renal transplantation. And then the second project I want to present today is the so-called EBV zero array project, which we work on in collaboration with a company in Berlin, and which is supported by the Investitionsbank Berlin. We are very glad that we got here support for this type of research, and we also got some support from the German self-help group Fatigatio. So to understand what we look for, I need to go a little bit into detail how the immune response against viruses looks like. So you are born with just naive B cells. With B cells, so these B cells have antibodies on the surface, and they are specific for all different types of infections you may get during life. And if you get an infection with EBV, then these B cells mature, and they mature into so-called plasma cells. These plasma cells, which make IgG, usually sit in the bone marrow, and they sit for many years and produce this IgG, which you can detect in the serum. But what is equally or even more important is that you also produce so-called memory B cells. These memory B cells do not secrete antibody. They still have immunoglobulin on the surface, and they usually sit in the lymph nodes, but you can also detect some of them in the circulation. And these are very important because plasma cells can live only for a few years, and then you need to send new memory B cells into the bone marrow. And we know now from many animal models that for a really good and protective, long-lasting immune response, you need these memory B cells. And I show you now some of the data we got from analyzing our patients with CFS. So first of all, we look at the antibody responses. We tested specific antibodies against this surface protein, VCA, IgG antibodies. Here you can see the levels of antibodies of healthy controls and of patients, and they look the same. But a subset of patients has IgM antibodies, that is approximately 10%. In addition, we made the observations that approximately 10% of patients do not mount antibodies against EBNA. And here as a control, we have also analyzed CMV antibodies. Both IgG and IgM look different look the same in patients and controls. So next we studied the memory B cells, which we can also measure from peripheral blood. We need to activate these cells that they make immunoglobulins, and then we can also measure this with a technique, an ELISA technique similar to the one we use to detect antibodies directly. And what you see here, you see all memory B cells. So we measure the total IgG producing memory B cells, and that looks pretty much the same in controls and patients. And then we look at specific memory B cells here against HSV type 1 virus, the lip blister virus, and against cytomegalovirus, that's another herpes virus, and it looked the same in patients and controls as well. And then what was quite striking is when we looked at the memory B cell response against Epstein-Barr virus, we found that against total virus, that's a lysate of EBV, and as well against proteins, the capsid protein against the EBNA protein, the number of memory B cells was in a subset of patients absent, and in many patients lower than in healthy controls. So it's approximately half of the patients who have diminished diminished memory B cell responses against EBV. So what does this mean? And possible explanations for this observation is that either they have mounted a deficient or insufficient response due to a late EBV infection, because that is well known that the later in life you get an infection, the less efficient you can make a long-lasting immune response. That's not only true for EBV, that is true for other diseases as well. That's the reason why we vaccinate our children very early, not because the later we do vaccinations, for example, hepatitis B, when we do it later in life, it's much more difficult to mount a long-lasting immune response. And we all know that this is very efficient, because if you vaccinate a baby against measles, it has lifelong protection due to the memory B cells you induce by repeated vaccination. The other possibility why memory B cells are diminished might be that you have used them up. We know from other chronic viral infections that there may be an exhaustion because you have not endless numbers of memory B cells. If you chronically are infected, you make a lot of antibodies and the plasma cells need repopulation by memory B cells. In the end, you have used them up and there are no naive EBV-specific B cells anymore, but you can produce new memory B cells. So both hypotheses are possible. However, there isn't much evidence that you have active EBV infection ongoing all the time in so many patients. There is a subset of patients, approximately 10%, where we have evidence of elevated EBV. Here we looked in peripheral blood immune cells. These again are controls. These are patients. These are not that high in numbers of EBV. So if you look in a patient with kidney transplantation, you are maybe that high. But we have evidence, and if you look in more detail in the B cells where the EBV virus sits, it looks similar. Interestingly, there are also some patients who have EBV in their non-B cells, which is already somehow abnormal. So we do not understand at the moment why this is. And the standard assay to study EBV load is to look for EBA, which is a very early produced gene in latent infection. But if EBV infection is active, if a lytic virus is made, then a lot of other proteins need to be produced as well. And we checked EBNA and we checked also this so-called BCLLF protein by PCR, and we have no evidence that EBV is lytically reactivated in all the patients we studied. This is a control the positive cell line, and this is in accordance with studies from other groups. So coming to our second project, the error array, a serum peptide array, what we do here is we measure the antibody response against EBV in much more detail, because you do not mount just two types of antibody, you mount antibodies against all different proteins of EBV and they're against different parts of the proteins. So what we do here is we measure the response against more than 2,000 peptides from EBV. So we test not the whole protein. That is quite difficult to produce a recombinant protein. Instead of proteins are made out of long chains of amino acids, and these you can synthetically produce. So you make 15 amino acid long chains and use these for testing. So-called 15-mer overlapping peptide libraries. That's what the company we work with is producing. And then these peptides are just coded on slides and are incubated with Serum. And then you get, of course, a lot of data which you need to automatically evaluate and which you need to analyze. So we need to do a lot of statistical testing. And this shows you an example how this looks like in the end. So each dot is a serum, and each of these plots is one peptide, and there you see several sera not responding. Here is a positive response, and again here are several positives, and this compares here healthies and patients. And then you see peptides where you have no response at all, and you see areas where you have some responses. And in the end it looks like this, so-called heat maps. Yellow or white means no response, and the stronger the response is, the redder the signal gets. And this is the peptides in total we studied. These were more than 5,000 peptides, of the 2,000 from different EBV proteins, and in addition various other infectious organisms which have been discussed to be probably associated with CFS and also some controls like influenza. And I will go with you through some examples now of what we've observed. This shows you data from two arrays. In this one, we tested healthy people, and each row is the response against one single peptide in a group of healthy people, or here patients with depression, and that looks pretty similar. We also tested patients with MS, where we see a much stronger response, especially against certain regions. This is patients with CFS, which also looks in certain areas different to healthy, and patients with depression. Another control is patients with Hodgkin disease. These are all patients who got cured from Hodgkin disease, but in cancer, you may know that there's this disease which is also quite severe, the so-called tumor-associated fatigue, which has similarities to chronic fatigue syndrome, but also differences. And here it looks almost the same, the response with and without fatigue. So this suggests that this has nothing to do with EBV. Then this is an interesting group. These are patients with lupus erythematosus. That's a disease where we know that it's associated with EBV as well. And in these patients, we can usually detect active EBV most of the time. And that corresponds that we have high signals against many of these peptides. And these were sera we got from Professor Flugge and Meller from Bergen, and also includes patients who were treated within the rituximab trials. So here I show you in more detail the data comparing the responses in CFS patient and healthy people. And here are only shown the peptides where we have a significant difference. And we usually see stronger responses in the patients as compared to control. But it's never black-white. And that's something we wouldn't have expected. As Maddie already said, it's a heterogeneous disease, CFS. I think that's pretty clear. And in addition, the type of antibody you mount is also dependent on your HLA composition. So not everybody of us makes the identical type of antibody. Yeah. 2 more minutes. So just to briefly summarize, I wanted to show you that most of the different responses we saw against EBNA proteins, this here is the EBV virus, and the EBNA1 is an especially interesting of these EBNA proteins because it has this weird sequence of 70 amino acids, which are just composed of 2 different amino acids, glycine and alanine. And this area has been known already that it's an area where antibodies in MS patients are found, whereas healthy people have no antibodies against this region. And that could be confirmed by our sera array as well. That's important for us, that we see that this assay is telling us things which have been known already. So this is patients with MS, with healthy people. If you want to know into this, if you want to look into this in more detail, this is recently published. And in lupus, we could also see strong responses. And lupus is very interesting, again, in regard to EBNA1, because there is also a mimicry. So patients with lupus respond to a certain area in EBNA1 very strongly, and this cross-reacts with a structure which is known to be an important autoantigen in a subset of lupus patients. So to sum up, we observe different response patterns against EBV in different patients' cohorts. We have enhanced responses against several peptides in CFS compared to healthy controls. That was reproducible in a second array. We saw responses against overlapping peptides. That means there are really regions where we detect enhanced responses. We observed different patterns in Germany and Norway, which may be related to some heterogeneous MHC background. And interestingly, we also saw a different pattern in rituximab responder and non-responder. Little difference in CFS between healthy and depression, no difference in tumor fatigue. And these assays are hopefully a basis now for diagnostic tests and treatment development. And we work with this company and hope we can come up next year with the first ELISA tests, and these are all the people working on this project, and I want to thank also the Selfhelp group for supporting us.
Thank you, Carmen. Couple of questions? Yes, sir.
Or subset of patients who have poor memory responses to EBV, do you think that group may be amenable to the ex vivo expansion of their T cells against EBV protein and then giving it back to the patients?
That's an interesting idea we had as well already. You may know that there are some reports on treatment of patients with MS, and there was a previous study from our institute more than 10 years ago where the patient with active EBV infection was also treated with such type of T cells. They had a full-blown chronic fatigue syndrome. We do not know yet if these patients with the memory B cells are the same who have diminished T cells. This is all work in progress, but this is one way we think of. And there was a recent study in MS showing that EBV-specific T cells were effective in 3 patients who got adoptive T cell therapy with EBV-specific T cells. Very recent study, small study. If you haven't seen it, I send you the link.
Okay.